So the models I just showed you are quite simple and make lots of obviously overly simplistic assumptions. So we use some individual-based simulations to simulate some more complex and realistic scenarios, and importantly, these account for genetic drift, demographic stochasticity, varying initial allele frequencies, and mixed genetic architectures. And we model a large mammal life history where we have low fecundity and high survival, so a high probability of an offspring surviving to reproductive age. So in the individual-based simulations, we have a polygenic architecture where there are 100 small effect loci with equal effects, and we model beta-distributed initial allele frequencies. With the mixed genetic architecture, we have 99 small effect loci, one locus being responsible for between 50 and 90% of the genetic variance, and the rest of the genetic variance spread equally among the 99 small effect loci. The initial large effect allele frequency is modeled between initial allele frequencies of 0.05 and 0.95. We assume carrying capacity is 1,000 and the initial population size is 500. And we ran 500 simulation repetitions for each genetic architecture. And then we tracked the trait mean and the population size through time. What I'm showing you here is on the y-axis is the mean phenotype, on the x-axis is time in generations. The results from the polygenic scenario are shown in orange, the major locus or large effect scenario is shown in blue. And we can see that in the polygenic case, the populations are more able to closely approximate the new phenotypic optimum than when the trait is affected by a major locus. There's also a lot more variance in the eventual mean phenotype among simulation repetitions with the large effect locus. And I should say the means among all 500 reps are shown with the thick lines here. This is the population size versus time for both genetic architectures, and we can see that on average, shown in the dark lines, that the population size is larger on average in the polygenic scenario than with the presence of a major locus. And here I'm showing you the proportion of populations that went extinct on the y-axis versus time on the x-axis. And the blue is showing the proportion of populations that went extinct when there was a major locus present. And the proportion of populations that went extinct is about twice as high when a major locus was involved compared to when the selected trait was polygenic. What I'm showing you on the y-axis here is the final population size, N80, versus the initial allele frequency at the large effect locus for each of the 500 simulation repetitions. And what this is showing is that there's a really strong effect of the initial allele frequency on the population dynamics. And it's beneficial, or the population viability and the eventual population size is higher when the beneficial large effect allele is initially rare. This is showing you the mean and 95% bootstrap confidence interval here. Here we have, or I'm showing the effects of the large effect size on population viability. So we have the proportion of populations extinct on the y-axis and generation on the x-axis. And I'm showing you the results for when the major locus was responsible for 50%, 70%, or 90% of the total genetic variance. And what this is showing is that when the large effect locus contributes a smaller proportion of the total genetic variance, the population dynamics begin to more closely approximate that of the polygenic scenario. So major locus effect size has a large influence on the population dynamics. And these results are showing that adaptation with a large effect locus is often too slow to prevent extinction. And we wanted to quantify this, and we asked the question, does the selection limit or the maximum ability of a population to evolve over the short term, does that really affect, is that really what's going on in driving the dynamics here? So we define something we're calling the short-term selection limit, and this is the expected phenotypic change over the first 10 generations, assuming that they're under idealistic conditions of no genetic drift and consistently strong selection, where the optimum phenotype is always 10 units higher than the current mean phenotype. And these are the results. On the y-axis here, I have population persistence coded as 0 for extinct. So these populations went extinct at 0. These populations at 1 persisted. And I fitted a logistic regression model for persistence versus the initial short-term selection limit shown on the x-axis. And the selection limit is a very strong, very good predictor of population persistence in this case. It's highly statistically significant. But if we fit the same model for the polygenic scenario, this relationship does not exist. There's essentially no relationship between population persistence and the selection limit. And I think what's going on here is that in all of these cases, there is just a lot more short-term adaptive potential associated with the polygenic architecture than when a major locus is present. And I should say that these findings hold under a very wide range of scenarios, including stochastic and temporally increasing phenotypic optimum, under phenotypic plasticity, with a smaller shift in the phenotypic optimum while holding the fitness function constant, under lower and higher heritabilities, and with larger and smaller shifts in the new phenotypic optimum, and with gene flow from populations with different phenotypic optima. And what does all this mean? I think the main finding here is that polygenic architectures generally confer higher adaptive potential and population viability compared to architectures with large effect loci. And I think it's important also to point out that just identifying trait loci, say in a GWAS, and identifying those loci that have statistically significant effects on a phenotype of interest is really insufficient to predict the selection response. And this is due to much of the genetic variance being unaccounted for inevitably by those loci detected in association analyses. And I think this quote from Ruth Shaw in her very nice paper in 2019 quite nicely summarizes this, where she challenged the idea. With current heavy investment in genomics and other molecular tools, some have opined that those approaches can effectively yield predictions of evolutionary adaptation in the sense of change in mean fitness. And she challenged this idea. And she went on to suggest that purely classical quantitative genetic approaches will be more efficient at predicting adaptation to things like environmental change than purely genomic approaches. However, I think our results also challenge that idea. And our results suggest that accounting for all of the genetic variants, including that portion of the genetic variants that we can't detect in association analyses, is really important. And we must also account for the strong effects of the genetic architecture of a selected trait on the temporal dynamics of heritability. And doing so should improve the prediction of population responses to environmental change. And I think that what all of this suggests is that what's really necessary is to integrate findings from modern genomics, in other words, loci that we find that are associated with traits that are under selection, into classical quantitative genetic analyses and population viability analysis. And we believe that this is likely one of the best ways forward towards an understanding of how populations will respond to environmental change, like global warming. I'd like to thank my collaborator, Gordon Lukart, and our funders. And I'd also like to thank the organizers again. And I really appreciate you listening. Thank you.